next we have Ruth Carmicheal. She is a senior marine biologist at Dolphin Island Sea Lab and a professor of marine sciences at the University of South Alabama. She is a population and trophic ecologist who uses traditional ecology techniques coupled with elemental and telemetry methods to understand how organisms respond to environment disturbances from physiological responses to changes in growth and survival or movement patterns. So go ahead and thank you very much.
All right, thank you. All right, so, a majority of work that's been done, spill-related research, similar to the research that you just heard about, especially early on, was really highly focused on direct toxicity to animals like you see here, and also on microbial degradation of oil. And our team was interested in really sort of what happened next, or what happened outside of direct toxicity. So did oil enter the coastal food web, and in particular, was this potentially an alternative pathway for degradation or breakdown of oil and elemental cycling? And so this includes oil, not only the crude oil that was coming into the system, but also the weathered oil on the surface, and tar balls and tar patties that we frequently saw washing up on beaches, and in some cases, continue to see. And we were interested then if organisms like oysters, for example, are getting this, essentially this oil is a slug of organic material, and if it's being broken up into the water column and becoming small particles, is it then, when it's being assimilated, consumed and assimilated, does it then provide the potential for these organisms to be used as bioindicators of oil exposure and entry into the food web and mobilization at the ecosystem level? And so for a long time, organisms like oysters have been used as bioindicators of pollution, and NOAA's Mussel Watch program is a great example of that, bioindicator program. But most of these programs rely on soft tissues, which require regular, and often if there's a pulse event like something like the Deepwater Horizon oil spill, it would require fairly frequent sampling of those soft tissues to detect some kind of toxin or harmful change in the water quality. And so what our group and others have been working on in recent years is the potential for shell to provide information that might also be indicative of contaminant exposure, such as assimilation of oil-derived materials and elements from those oil materials. And so we've been looking at a number of studies that would allow us to gather data on the shell that might be then complementary for these traditional soft tissue analyses. So the work that I'm going to talk to you all about today is a combination of field study that was done along the Alabama coast, and partially was taking advantage of the fact that we already had oysters out in cages along the coast, and so we could get data from these animals for periods before, during, and after the spill. And then also we followed up with controlled laboratory studies where we exposed oysters, young oysters, to a variety of different types of forms of oil, similar to what you heard about in the last presentation, where we were looking at crude oil, also different forms of weathered oil, as well, the tar balls and tar patties. And then we also went and sampled native oyster reefs along the coast in Alabama, Mississippi, and Louisiana that are part of the Nature Conservancy's oyster restoration program. And then in all these cases, we looked at both the organic elements that would be assimilated and sort of captured in the organic matrix of the shell. And we also looked at inorganic elements, specifically trace elements or trace metals that would then be incorporated into the shell. So we could look at both the organic and inorganic assimilation. And then we also considered factors such as the oil concentration, and also, particularly for our area, salinity, which is very important due to low salinity stress, and also for the reasons that were just mentioned in the previous presentation, that there was freshwater diversion was used to remove or move oil potentially out of the system, and had some salinity effects. So we were looking at salinity as a potential stressor. And then we also wanted to compare the values that we're finding in tissue to shell to determine how well shell might be actually acting as a proxy for tissue, and whether it was directly reflecting what we would expect to see in tissue as well. So we had a whole lot of collaborators and co-authors involved in this work, and a number of graduates, interns, graduate students, and partners. But I'm going to highlight specifically the work that was done by students in my lab, understand that there's a whole lot of other people who are also involved in this, and you can certainly find them as on our papers. And then I also wanted to just point out the approach we used for the trace element analysis was a combination of laser ablation and solution-based and inductively coupled plasma mass spec. So this kind of gives you an idea of what we were doing. So our first study, which is the field study along the Alabama coast, is published in Environmental Science and Technology, and I encourage you to go grab that paper. The two students in my lab, Dr. Heather Patterson, who is now a postdoc at the University of Alabama in Birmingham, and Amanda Jones Knobloch, who was an undergraduate intern at the time and is now also a postdoc. So time flies, it's amazing. But the work that we did was at these sites shown here in the map along the bottom, and we know that there was potential for our sites to be exposed at various times to various levels of oil throughout the spill. And basically what we did was we used hatchery-reared young oysters, not the juveniles that you heard about in the previous presentation, but a little bit older than that, a little bit bigger, the kind you might put out for just starting for aquaculture. And so we had before shell samples. And then for the samples during and following the spill, we actually shaved off the known growth during that period, and we just analyzed that new growth. So we could sample pre, during, and post-spill. We could sample those in the same animals. And so what I'm showing you here is looking at that organic matrix of the shell and looking specifically at carbon and nitrogen that was assimilated in that organic matrix. And we also, for comparison purposes, and these are mean values for all of our sites, we looked at the weathered oil that we found at those sites. Weathered oil had very distinctive nitrogen and carbon signatures where the nitrogen was quite light. And then the carbon, and this is consistent with what other researchers have found, is consistently right at this -27, which is actually reflective of terrestrial carbon and similar to freshwater land-derived sources that we see coming into our system, not just from oil. And then we also looked at the suspended particles, that's the SPM, that was basically the ambient available food that's floating around in the water column for the oysters at those sites. And then we looked at the organic matrix of the shell itself, which is the part that's shown in the larger circle up here. And when we look at these and with the known expected difference between what these animals are eating, what they're actually consuming, and what their values in their shell should look like, these animals are clearly eating something more similar to this ambient available diet and not showing clear consumption of oil-derived elements, a mixture of these suspended particles and oil. It looks pretty clear that they're primarily eating that ambient background food supply. So that's just suggesting to us, at least in the organic elements, that we're seeing they're eating their ambient, marine phytoplankton-based food supply with a little bit of freshwater influence as opposed to oil-derived materials. Then when we looked at organic elements, so these are the trace metals, and I'm just showing you one portion of our data here, and it's the elements that are, of the 26 that we looked at, those that are typically most associated with oil exposure. And so what you're looking at is the trace element concentration compared to that original hatchery shell and then that newly laid down shell at each of our different sites here. And what we found was overall post-spill, we found greater elemental assimilation in the new material compared to during spill. But there were certain elements, in this case lead and vanadium in particular, that seemed to be assimilated at higher concentrations than the hatchery shell. And so this indicated to us that the trace elements may be better indicators of oil exposure than the organic elements that are primarily reflecting the background diet. The other thing that we noticed, of course, is that in the hatchery shell from animals that were just reared out along the coast prior to the study, you do see assimilation of elements, a variety of elements that are associated with oil, and the values for even the vanadium and the lead are in the range of what we're seeing in the new shell. And so this suggested that there could be a problem with just high background in these systems anyway that would make it difficult to detect an oil-specific exposure. And so this left us a little bit with some inconclusive data to understanding whether or not we were really getting just an ambient background signature or whether we might be getting a specific oil exposure. So we followed this up with the controlled lab studies. And as I described to you, we exposed these oysters with a WAF, the way that was described in the previous study, with various types of oil. We exposed them for 16 weeks. with 2 different concentrations of oil. And we also included background water from Mobile Bay up in the industrialized northern part of the bay so that we could specifically target this potential for a background signature. And then we also looked at 2 salinities that were common during the spill. But anyone who knows our area, it even gets more stressful and lower salinity than this. And this just shows you a picture of our setup. And here was led by Kimberly Peter, who just recently received her master's working on this work. And then the second part of this was going and sampling those native Nature Conservancy reefs along the coast to look at what those adult native oysters out in the field actually looked like elementally. And so I'm going to give you the 40,000 foot sort of perspective on this because we don't have time to go into all the detail of all the cool results. But just to kind of summarize it, we found really importantly, low salinity and higher oil concentration did depress growth in the juvenile oysters in that lab study. And we found that both salinity and concentration affected the assimilation of individual elements. And so we found that with lower salinity, we actually tended to get enhanced vanadium assimilation, but depressed zinc. And the higher concentration of oil actually appeared to depress the assimilation of cadmium, but other elements were not different. So that was kind of interesting. We saw some things that we didn't necessarily expect to see that show us that this may be a little bit more complicated a story. We also did find that the trace elements in the animals that were incubated in the Mobile Bay water were very similar to our treatments. So corroborating the fact that we do have potential for a high background of elements in the system from a variety of anthropogenic sources that may confound the ability to detect an oil-specific signature. And then we also found seasonal, and I'm not going to show you all these data, but I've just picked out some of the regional differences here where we found higher, again, vanadium in Alabama and Louisiana compared to Mississippi. And this is consistent with also what we know about the shoreline land use and industrialization in the areas adjacent to these reefs that we sampled. And so this makes a lot of sense that we're picking up just a higher anthropogenic signature in general at some of these reefs than others. And then we didn't find, though, even though we found these differences in assimilation of some elements regionally, there was no difference in condition or growth regionally. So if there is an effect of variation in this elemental assimilation, it doesn't appear to be having an effect on their growth or condition. And then finally, to look at the tissue shell comparison, this is work that was just done this past summer by Tiara Turner, who is a research experience for undergraduate student from Judson College here in Northern Alabama, and Audrey McQuigg, who's been an intern in my lab for a few years and recently just matriculated as a student in our program, a graduate student. So we're excited to have her on board. So what these guys did was they took tissue and shell from the same critters, and they did the analysis on the elements and then compared them directly to each other. So you're looking at the ratio of the concentration in tissue compared to shell for those adult native oysters at the reefs, and then below that for the lab animals at the lower and higher salinity. And what they found was that the values in the shell followed the values in the tissue. So as you saw higher values on average in tissue, you also saw higher values in shell, which was very interesting. Something else we found that was particularly interesting is that there tended to be certain elements that were consistently higher in shell and others that were consistently higher in tissue. But there was this suite of elements that appeared to be assimilated very close to one-to-one. And in particular, magnesium and again, our friend vanadium, which is popping up repeatedly, were actually the only elements that were conserved and seemed to be assimilated at one-to-one in tissue and shell in all of the treatments. It didn't matter whether we were looking at the adults, the younger animals, whether they were higher or lower salinity. These elements seemed to be pretty well conserved, which was interesting and suggests that they might be quite useful as indicators. But we also did see that salinity again was a source of variation, where we saw overall greater differences between tissue and shell at the lower salinity. And so we saw them more falling off that line and showing greater variation. So what does all of this mean? Well, in terms of a big picture take-home message, the shells of oysters and other bivalves, we're getting this growing body of evidence that show us that they can be used to reconstruct these longer-term or historical pollution events, even in cases where soft tissue, it might not be feasible to get soft tissues, or maybe animals have died and only the shell is left behind. So there's a lot of potential for this method with some caveats. So particularly with regard to the Deepwater Horizon spill, and I suspect this is probably true in a lot of cases, when we're looking at the organic component of that shell that's trapped in that matrix. We didn't find any evidence of assimilation of oil-derived carbon and nitrogen. That seemed to be reflective of the overall background diet. So we're just not, if there was assimilation of oil-derived elements, we're just not picking it up against the background. The other issue that's particularly important in our area is that that oil-derived carbon has a terrestrial signature, and we have so much fresh water coming down delivering land-derived carbon to our system that it would make that difficult to detect very likely anyway in a system like ours, but it may be easier to detect in a system that has less freshwater input. And then trace metals, they seem to be potentially more useful for specifically picking up oil exposure or anthropogenic inputs in general of elemental contamination. And we were able to detect seasonal and regional variation that suggests that this actually might be useful for doing essentially like a shoreline mapping to see whether or not areas of high particular types of land use are actually resulting in an increased delivery of trace metals to the system. And then of course in our system, we do have to be concerned about high background, not only that terrestrial carbon that I just talked about, but high trace metal delivery from any of the anthropogenic sources that we have in some of these not only industrialized ports like Alabama Port and coming down the Mississippi River, but also what's coming down in the fresh water being conveyed to these systems. I mean, we have the Mississippi River and then Mobile Bay, the fourth, or depending on the data you look at, the third or fourth largest freshwater drainage among watersheds in the United States. So we have tremendous amounts of land-derived elements and carbon coming into our system from sources all around the watershed. And so it may make it difficult to pick up a specific point source signature in that delivery. And so this system actually is probably one of the most difficult systems in which to test and apply this approach. And so the fact that it worked for us is exciting, but it may even be more successful in systems without so much complexity. And then as far as shell being used as a proxy for tissue, we found that surprisingly, there's a lot of consistency between tissue and shell that suggests that in some cases it can even be used as a direct proxy, but because of that consistency, it probably can be even corrected for and used in that way. But what we have to really pay attention to, and I think this is my big message for you all from the suite of our data, is that the factors that are affecting the delivery of elements such as freshwater discharge are also affecting environmental attributes like salinity, that is then affecting the bioavailability. It's changing the chemistry of the water, not only determining what elements are there, but what forms they're in and whether they're available to be picked up and used by organisms in the system. And then that same environmental stressor is a stressor on oysters and their physiology. So if they are too stressed out to filter and feed, they're also not going to be able to take in and assimilate these elements and act as bioindicators. So we have to consider all of these complex factors together. And one of the big points I want to make, and this is from work that Dr. Patterson did in my lab, is that in the Gulf of Mexico, our oysters are stressed anyway. So this shows you relative intensity of stress proteins, and they are upregulated when dissolved oxygen concentrations are low, but that stress decreases as concentration of oxygen goes up, except when salinity is low. So when salinity is low, even when dissolved oxygen concentrations are high, these animals are still stressed out. And so the moral of the story with respect to an oil spill is that if you have something like the Deepwater Horizon oil spill where there's this delivery of organic matter, we have increased microbial activity that's drawing down dissolved oxygen from the water column. And then we do something like we either have high freshwater inputs or we're opening sluice gates to push the water away. We may be actually subjecting these animals to higher, just normal background stressors that may be as harmful or more so than the oil itself, at least in terms of, for these animals that we're talking about, that are a little bit older, and we're talking about changes in water quality. We just want to be sure that the cure isn't worse than the disease. And we want to make sure that we're understanding what we're doing in the context of the system and how we're interpreting these data. So with that, I will stop. I see that you're up telling me to stop. And I thank you all very much for listening in, and also to all of our funders that helped support this work over the last 10 years. So thank you.
Wonderful. Thank you very much. We appreciate it. That was great.